Okay, everybody, good morning or afternoon or wherever you are. Welcome. We're happy to see participants coming in. Um, Welcome to our second public webinar on RBE. We've had several private ones. These are for TG 118, looking at RBE weighting factor and quality factor. And we're really excited because we have two teams, I guess, or two groups of speakers. The first speaker today I'm going to introduce right now is Igor Shuryak. Igor is from Columbia University. He's going to present work that he and David Brenner have been doing, and David is also here kind of as a backup to discuss and answer questions that might come up as well. The way we're going to do this is we're going to allow each speaker to talk. We'll then have questions that can be submitted in the Q&A, or if any of the panelists want to ask, they can actually just raise their hand and I'll call them. We'll allow each of the speakers to speak, and then we'll handle Q&A at the end for both of them. Welcome, everybody. Igor, if you're ready to start, I'm going to introduce you. Igor Shuryak from Columbia University, as I said, along with David Brenner, and he's going to talk about quantifying relative effects of two radiations independent of their dose-response shapes. Igor, please.
Yes, thank you very much. So I will share. So I'm very much honored by the invitation to speak today. And as we said, this work is guided also by my mentor from Columbia, Professor David Brenner, and also Professor Sachs from Berkeley, and Albert Fornace and his team from Georgetown University. So for the foreseeable future, we expect that scaling human cancer risks from low LET radiation to high LET radiation will be based on scaling factors that are determined by animal studies. And this might work by like something like this, that mice or other animals are exposed to gamma rays and to heavy ions or other radiations of interest, and scaling factors are determined from this work. And then these factors will be applied to scale risks from gamma rays to high LET ions in humans. But of course, how actually to do this conversion and scaling is not a trivial task. So the current way to do it is RBE, which involves calculating a ratio of doses which produce the same effect. So effectively, it is a horizontal scaling of dose response curves. But also there is an alternative concept of vertical scaling, where you take the ratio of effects of different radiations at the same dose. And these two concepts are shown by these schematic diagrams on the left and on the right. So for example, here, x-axis would be dose, and y-axis is some time of response, for example, tumors per animal. And there is a dose response for gamma rays and a dose response for high LET radiation of interest. So the RBE concept involves scaling like this, taking, say, an effect here and looking for comparing the doses. So what dose of gamma rays would give the same effect as this dose of high LET radiation? And the vertical scaling concept is different, that you pick a dose of interest and compare vertically these arrows. This would be the effect for high LET, and this is effect for photons. So the RBE concept historically is tied to the cancer radiotherapy field, because there it was very important to identify isoeffective doses of different radiations or regimens that have the same anti-cancer effect. However, in radiation risk estimation field, the isoeffective dose concept is not as relevant, because the focus is on risk magnitudes and predicting them. And for this reason, this alternative idea of using vertical scaling instead of horizontal was considered actually by many researchers. And for example, by Dr. Sabatier in France, and they published a paper which is called Comparison of Individual Radiosensitivity to Gamma Rays and Carbon Ions. And they said here, we define a new ratio called RDE, calculated by dividing the mean DSBs per cell, which was their endpoint, following exposure to carbon ions. By that, following exposure to the same dose of gamma rays. And this differs, of course, conceptually from RBE. And these graphs at the top are taken from their paper, where these are the dose responses for gamma rays and for carbon ions for their endpoint. And these are linear quadratic curves. And the ratio of the effects has a dose response that looks like this. So they calculated vertical scaling for monotonically increasing linear quadratic dose response shapes, which are appropriate for this endpoint like DSBs per cell. But here we talk about generalizing this approach to other dose response shapes, including those that saturate or decrease at high doses, which is relevant for cancer, for example. So again, looking at this type of schematics, you can see that horizontal scaling can run into problems in some cases, like for example, this one. If the dose responses flatten or they start to decrease at a high dose, for example, here, there is no corresponding dose of gamma rays, which would give the same effect as this dose of heavy ions, because the responses turn down or flatten. But the vertical scaling concept doesn't really have this problem. For example, here you can still calculate the ratio of effects. And also, in cases where the dose responses are linear, then these vertical and horizontal scaling concepts are actually the same. So in this case, we used RER as the abbreviation for radiation effects ratio. But if they're not linear, then they're not the same. And recently, we applied this concept to a system from our collaborator, Dr. Fornes at Georgetown University, who used an APC mutant mouse system to find tumors induced by space-relevant doses of protons and various types of ions, up to very heavy ones, and also gamma rays. So this was published recently based on this work. So the modeling approach to model the dose response shapes, which are nonlinear, was here where this M is the dose response function, B in black is the black background tumor parameter, and the blue terms are for non-targeted effects of radiation. And here you can see that they are non-linear with a plateau term and also an exponential type of slope. So this saturates at higher doses. And the red part is a linear targeted effects contribution with a slope. And also we considered a green cell killing term for turn down, potentially at high doses, but it was not supported by the data that we used here. And also it was found that the tumor count data per mouse were under or over dispersed relative to the Poisson distribution. And this varied depending on radiation type, or for example, what type of ions, and also on the dose. So to model this, we had to use different distributions, and we used this weighted negative binomial distribution shown here, which has two extra parameters called R and Q, which describe the variance, which can be less than the mean or greater than the mean. And then the mean number of tumors is given by this function here, where M is from the previous equation. And the general behavior of this model is something like this schematic, where there are two components contributing to the total response. So the targeted response component is linear as function of dose, but the non-targeted component is not linear. It saturates. So the sum, which is the red curve, has a nonlinear behavior, especially at low doses, and approaches linear at high doses. And these graphs show the actual fits of this model to the mouse data, where each panel is a different type of space radiation, with the LETs also written for reference. And x-axis is dose, and y-axis is tumors per mouse. So the red lines are the fits of this model, and the blue dots are the data for individual mice, and the black squares and dots are the summaries, the means and the standard errors. So as you can see, especially for these heavier ions, there is definitely a nonlinearity, a bump of some kind at low doses of around 5 or 10 centigrade here. And this is attributed to the non-targeted effects by this model. And this is a continuation for heavier ions, silicon and iron, which were the most effective, and also for gamma rays for comparison. And again, you can see this even more nonlinearity and more dispersion for the heavier ions and much straighter for gamma rays. And here is a magnification for convenience of the gamma ray response and the silicon ion response at the low doses of 5 and 10 centigrade and 0. So these were randomly jittered here so you could see more clearly the distribution. And of course, for example, at these doses, the ions have a bigger mean response, but also much bigger dispersion than the gamma rays. And then based on these fits of the model and their uncertainties, we can calculate the RBE and the RER metrics. So here again, each panel is a different radiation type, and y-axis here is RBE relative to gamma rays, and x-axis is dose, and both the y and the x-axis are shown on the log scale to clearly see what the predictions look like from a low dose to a high dose. Of course, at the low dose, there are extrapolations. So these are based on the model fits. So red lines here are the predictions, and the dashed lines are the lower and upper 95% confidence intervals. So as you can see here, the RBE is dose dependent because of the nonlinear shape of the dose responses, especially for ions. And it can reach high values, more than 10 or even up to 30 or so, especially for the silicon and iron ions. And this occurs in a dose range of, say, a few centigrade, which is quite relevant for space. And then for comparison, these are the RER. So these are the vertical scaling ratio estimates of the dose responses. And these are somewhat different and generally lower than the RBE. And they're also dose dependent, but less curved than the RBE estimates. And also highest for silicon and iron ions, but here the values are between 5 and 10. And this schematic shows the general differences between using these concepts, the RBE concept and the vertical scaling concept. So here are hypothetical dose responses, for example, for gamma rays, something like a linear, and for the heavy ions, more like a concave curve. So the main difference is that to get a good RBE estimate, you need to have relatively good knowledge of the dose responses, of the dose response shapes. But for RER, potentially you don't need to have as much knowledge of the dose response, because if you're interested in the risk at a specific dose or dose range, which is marked, say, by gray here, you just need a gamma ray estimate and an ion estimate, and you can divide one by the other to calculate the risk. So in a summary, the RER uses an alternative approach for scaling human risks from low LET to high LET based on animal data. Unlike RBE, this concept compares the low LET and high LET risks at the same dose, so it scales vertically. And unlike RBE, it does not require knowledge of dose response shapes. And potential advantage of this is that fewer radiation doses need to be studied in the animal carcinogenesis experiments. And also, for example, because of the influence of non-targeted effects at low doses causing nonlinearities, in our analysis, RER is often smaller than RBE. So actually using RER instead of RBE can potentially result in smaller high LET human cancer risk estimates. So thank you very much once again. This is it for my slides.
Okay, thank you so much, Igor. And yeah, as I said, we're going to do both presentations and then allow for questions. So please, already you can start entering your questions for Igor and David in the chat. Um, so let's go on to our second speaker. So Tatsuhiko Sato, who is from Japan, he's going to also present this morning, and he's going to talk about RBE for skin reaction, review of past experimental data, and proposal of a new model. Tatsu, please, why don't you share your slides?
Okay, thank you for introduction. I would like to talk about the RV for skin reaction, review of the past experimental data, and the proposal of the new model. So I'm extremely happy because our paper, including the most contents of this presentation, was just published in Red Journal, actually yesterday. So it's quite new data. So if you are interested in this topic, so please read this paper. Of course, questions or comment after this presentation are very welcome. So here is a table of contents of my talk today. And first, I would like to talk about background purpose for the necessity of evaluating RV for tissue reactions. As for the radiolocus protection purpose aspect, in ICRU report 95 published in 2020, it is written that limits to prevent damage to lens of the eye and local skin should be set in absolute dose rather than the equivalent dose, because WR used in the calculation of equivalent dose relate to stochastic effect rather than tissue reaction. I fully agree with this, but how about the case of the mixed radiation field? So such as emergency exposure due to nuclear accident or cosmic ray exposure during large solar particle event. So far as I know, no answer has yet been provided. I think the evaluation of RV for tissue reaction is indispensable for establishing future radiological protection system, as is the same for stochastic effect. As for the medical physics aspect, in the treatment planning of carbon ion therapy, RV are evaluated by models such as microdosimetric kinetic model or local effect model. And their model parameters were generally evaluated from the surviving fraction tumor cells, and an empirical scaling factor is introduced to convert in vitro RV to clinical RV. However, several questions still remains, such as the adequacy of the surrogate use of in vitro experimental data for parameter determination in the models, and the assumption of the use of the same RV for therapeutic effect and normal tissue complication. Therefore, a possible link between in vitro and in vivo RV must be investigated. So let's look at the past evaluation by ICRP and NCRP. ICRP publication 58, published in 1990. Excuse me. Sorry. ICRP publication 58 reviews the past experimental data of RV for various tissue reactions and summarize overall ranges of RV in its table 6. I will show the table 6 in the next slide. And NCRP report 132, radiological protection entitled Radiological Protection Guidance for Activity in Low Earth Orbit. proposed a recommended RV for converting gray to gray equivalent for deterministic effect based on ICRP 58. And ICRP publication 92 entitled RV quality factor and radiation weighting factor discussed the linkage of RV maximum to cell killing. And it is also written in the publication that with regard to skin, it will be appropriate to use an annual occupational dose limit from the densely ionizing radiation 0.5 gray divided by a weighting factor based on the RV value from clinical or experimental experience against a low LET absorbed dose of 0.5 gray. However, no concrete value has yet been provided to for the RV value. So here is the past evaluated RV tables taken from ICRP 58 and NCRP 132. ICRP evaluates the typical ranges of RV for 1 to 5 MeV neutrons, 5 to 50 MeV neutrons, and heavy ions for single exposure or fractionated exposure or RV maximum. Under NCRP evaluates their recommended RV by taking the mean value of fractionated exposure in addition to their own evaluation for proton over 2 MeV. And as you see, the RV increase with a decrease of dose per fraction. And RV are generally smaller than the corresponding radiation weighting factor. So that means RV for tissue reactions are smaller than RV for stochastic effect in general. So, however, these values are mainly evaluated from the result of in vivo experiment performed before 1990. And one of the objectives of our task group is to review the determination of RV and especially derivative value Q and WR and provide support or lack of support for particular values and models and determinations that influence radiological protection. So to achieve this object, at first, we started to review the past experimental data for skin reactions and dermal cell survival. So why did we choose skin? The answer is very simple. So many experimental data are available for skin RV, and it's good for starting the discussion. So next, I would like to talk about the review of past experimental data. At first, we tried to find all experimental data cited in ICRP 58. And next, we searched RV plus skin or RV plus derma in PubMed, and approximately 220 articles were found. Then we selected the studies employed 2 types or more radiations and included sufficient information for reproducing experimental condition. And finally, 23 articles are found for skin reactions and 8 articles are found for dermal cell survival. Here is a list of data for skin reaction that we found. Sorry for busy slide, but roughly 1/3 of data were taken after the publication of ICRP 58. And in 1970s to 80s, neutrons generated from deuteron reactions were frequently used. And while after 1990s, carbon ions were frequently used because carbon ion therapy is getting popular since then. So here is an example of the measured RV as a function of absorbed dose. This graph is for carbon ion 200 MeV, 290 MeV per nucleon SOVP against cesium gamma rays. And this graph is for 16 MeV deuteron, beryllium, neutron against X-ray 250 kV, kilovolt. And experimental data are rather scattered even for the same radiation conditions. And if I put evaluated value ICRP and NCRP, so the ICRP and the NCRP evaluations are rather consistent even with the recent data. So here is the recommended value of NCRP, and this range is ICRP evaluated range. So here is the RV maximum, here is a fractionated exposure, and here is a high dose exposure. Actually, ICRP didn't specify the exact dose, but so I roughly write the range from above 10 gray. So here is a list of data for dermal cell survival that we found. So all data are taken after year 2000. So I may find older data if I searched by other keyword, but this is all we could find at this moment. So here is an example of measured dermal cell surviving fraction for X-ray and carbon 135 MeV per nucleon SOVP beam. So if we calculate the RV for a few gray irradiations, it will be approximately 1.6 for front edge of the SOVP and 3.5 for the distal edge of the SOVP. And both are consistent with the ICRP evaluation, which are 1 to 4 or 2 to 3 gray per fraction of heavy ion radiation. However, it is hard to derive any concrete conclusion just from seeing the past experimental data. So we therefore propose a new model for comprehensively analyzing the in vivo and in vitro RV in the same framework. And next, I would like to talk about the model development. So here is a basic feature of our model. So it is based on the microdosimetric kinetic model, so-called MKM, which is originally developed for calculating cell survival using the LQ functions and currently adapted for estimating the clinical RV in HIMAC. And the two revisions were made for this application. One is the introduction of switching dose from LQ to linear functions. And the other is the reduction of the number of free parameters from 4 to 1. And the numerical value of the free parameter was determined from the least square footing of the experimental data by precisely reproducing the irradiation condition using the Pitts code. And not only the mean value, but also the uncertainty of the RV can be easily deduced from the model based on the standard deviation of the evaluated parameter from each experiment. And I will explain about each feature in the next few slides. So at first, I will explain about the theory of MKM. So basic assumptions of the model are as follows. First, cell nucleus can be divided into several sub-regions called domain. And two types of DNA damage named lethal and sublethal damage regions are created in cell nucleus due to the radiation exposure. And the initial number of DNA damages produced in the domain are proportional to the specific energy of the domain, but saturated at a certain specific energy. And the sublethal regions are to be repaired or converted into lethal region via spontaneous transformation or interaction with other sublethal region produced in the same domain. And a domain is considered to be dead when a lethal region is formed at the site. And a cell is considered to be dead when a domain in its cell nucleus has died. So in this case, only this guy survives. So under these assumptions, cell surviving fraction can be determined from the LQ relationship with only one quantity for characterizing the radiation field, which is saturation collected specific energy, G star, as calculated by this equation. And alpha beta for fundamental radiations and the saturation parameter and the domain radius are the free parameter independent of radiation field. Then we introduce three new assumptions. The first one is the saturation parameter Y0 is always equal to 100 kb per micron, because this is a rough value for the saturation where overkill effect occurs. And alpha beta, second one is alpha beta ratio for skin reaction is fixed to 10 for X-ray therapy radiation field, as written in the ICRP publication 118. And the function to express the dose dependence of surviving fraction changes from LQ to linear relationship at 7 layers. Then beta and domain radius becomes the only parameter for calculating surviving fraction. And considering the fact that the surviving fraction for both test and reference radiations are proportional to beta, domain radius RD becomes the only remaining parameter for calculating RV. So we have to evaluate this parameter. We can estimate the RV for skin reaction for any radiation period. So next I talk about how to determine the domain radius. And at first, we reproduce the irradiation condition of past experiment using the FITS code. And actually, this is the most difficult part of this study because these detailed experimental setups are generally not well explained in the literature. So we needed to imagine the experimental setup from the available information, such as constructing a virtual rich filter to reproduce the depth dose distribution of SOVP beam. Then we calculated the probability density of linear energy using the microdosimetric function implemented in FITS and find the best fit value of domain radius for converting the linear energy to specific energy for each study. And finally, we evaluate the mean and standard deviation of domain radius for in vivo and in vitro experiments separately and discuss the possible linkage between two RVs. So before going to the result of our model, I would like to briefly introduce our developing code FITS. So FITS represents a particle and heavy ion transport code system, and it can analyze the transport and collisions of nearly all particles over wide ranges. So that's why we adapted this code for this analysis. And it is all in one package type code. That means all contents of FITS are fully integrated in one package, and it is available in free of charge by submitting application form via FITS website. And more than 6,000 users obtain the code and use it for various purposes, such as accelerator design, radiation therapy and protection, and space and geoscience. So if you would like to know more about FITS, please visit our website here. Okay, so next I will talk about the results of our model calculations. This table shows the best fit domain radius to in vivo experimental data of skin reactions. And as you see, the best fit value here vary with expected. So their mean and standard deviations are 0.187 plus minus 0.043 microns. So this table shows the best fit domain radius to in vitro experimental data of dermal cell survival. And their mean and standard deviations is 0.251 plus minus 0.061 microns, which is larger than in vivo data with statistical significance. So larger domain radius result in the lower RVE in our model. So RV for dermal cell survival tend to be lower than that for skin reaction. So this is consistent with current high-mark treatment planning. They also assume a certain factor like 1.5 for converting the RV for cell survival to the clinical RV. And no other significant tendency can be observed for other analysis, such as I compared between the early and late effect of skin reaction, but the domain radius are quite similar. So this graph shows an example of the measured RV for skin reaction for carbon 290 MeV SOVP beam against cesium 137 gamma rays. And this is for 16 MeV barium, deuterium barium neutron against X-ray 200 kilovolt, as I showed before. And here is our calculated results. So the best fit to skin reaction is shown in the green area, and the best fit to cell survival are shown in the orange area. And the calculated result, particularly for the best fit result to skin reaction, agree with the experimental data fairly well. But some data are outside of the range because this shows just one sigma data. So of course, some experimental data outside of the range. And the comparison for all radiation conditions are presented in 38 supplemental figures in our paper. So please check them if you want to know more about the comparison. So this graph shows the calculated RV maximum and RV at 10 gray for monoenergetic neutron against cobalt 60 gamma rays. And as expected, neutron RV reaches maximum around 1 MeV due to recoil protons, feature approximately 8 for RV maximum and approximately 4 for RV for 10 gray radiations. And RV for thermal neutron irradiations are also high due to neutron capture reaction of nitrogen, which is specific to skin reaction. So in most cases, RV for thermal neutrons are not so high because gamma ray dose from the hydrogen capture are the dominant, particularly inside human body. So this graph shows similar data, but for monoenergetic photons. So as suggested by many experimental data, RV for low energy photons, such as kilovolt X-ray, are higher than the gamma rays. And this study can quantify the difference. So up to around 1.3 for RV maximum and 1.1 for 10 gray radiation. So this graph shows the similar data, but for monoenergetic carbon ions. So heavy ion data, including carbon ion data, as shown here, have a peak structure due to the saturation corrections. So The peak values are almost say independent of ion species because LET around 100 kb per micron always gives the highest value. And which is also around 8 for RV maximum and around 4 for RV for 10 gray irradiation. So lastly, I would like to show the comparison with past evaluation. Here is a table of ICRP and NCRP evaluations that I showed before. And red bars are corresponding data obtained from our calculation. So as you see, our calculations are rather consistent with the past evaluations. And this tendency confirms that the past evaluation made by ICRP and NCRP a few decades ago are still supported by analysis based on all-time experimental data, including recent ones. So let me summarize my talk. So past experimental data of RV for skin reaction and dermal cell survival were comprehensively reviewed. And a new model for estimating RV for any radiation condition was proposed based on microdosimetric kinetic model. And the model suggested that RV for skin reaction tend to be higher than that for dermal cell survival. And the past evaluation made by ICRP and NCRP are still supported by recent experimental data. So as for the future plans, similar analysis for other deterministic effect will be performed, and reproduction of the radiation condition is a key issue in such analysis. So if you are planning to measure RV, so please describe the experimental setups as detailed as possible in the paper. And in addition, the procedure for evaluation from the calculated result must be considered, such as how to simplify the data. Of course, our calculation is just calculation, so cannot be used for directly used for the evaluated value. So we have to consider about simplification. And also what those level is appropriate for representing the exposure scenario. So for stochastic effect, the RV maximum is definitely the best choice. But for tissue reaction, RV maximum is maybe too conservative, and we have to find certain dose to represent the exposure scenario. So lastly, I would like to introduce my collaborator on this topic, Matsuya-san and Hamada-san, and also our co-authoring paper again. And thank you very much for your attention.
Tatsu, thank you so much. This was really great. And there are already some questions in the Q&A. I want to invite people to ask questions of both of our speakers now. I'm going to start out at least with one question, allow the panelists first to ask questions, and then we'll go to the questions in the Q&A. Tatsu, I was going to ask you, you make this issue about the difference in the RBE between the dermal cell killing and the skin reaction. Why do you think that is? I wouldn't have predicted that.
Why?
Why?
That's a very difficult answer, particularly for me, because I am not a biologist. So I discussed with about that Hamada-san. He's an expert of radiation biology. He still think he has almost no idea why. But in general, that's my opinion. But I'm not a biologist. But like a more complex reaction, usually the RV values are higher. So like a cell survivals and the tissue reactions and like a stochastic effect. So the endpoint become more complex, the RV become more higher because a lot of biological system involved with the like a repair or carcinogenesis, or so that's roughly say I can say so, but that's my.
That should be in future plans, right? That should be in future plans. So,
Yeah.
okay, let me ask the panel. Is there anybody in the panel that wants to make additional points or make comments? I think you can just unmute yourself and do it. Anybody, any questions from the group before I go to the broad questions? Okay, so let me go to the questions in the Q&A, and if people want to speak up, they can. So I think the first one is for you, Igor, and it's got I guess a couple of parts, and it brings in Tatsu, your presentation as well. It's from Alan Calvert, and what he says is, it occurred to me several years ago with regard to childhood leukemia clusters in the UK and birth defects in Kerala that the statistical anomalies could be resolved by ascribing an RBE of 100 to 200 alpha radiation. Does anyone have any definitive epidemiological data that might lead to such a reevaluation? I don't know that either one of you can answer that, but let's go on to the follow-up. Thanks to Dr. Shuryak's graphs, it seems that RER could be very large for low doses of heavy ions and may be environment. What about ingested alphas?
Yes, so first of all, thank you for your interest, for your questions. I'm not sure I can completely answer this, especially the first part. In the data that we analyzed here, of course, the more data that accumulated, the more accurate we could be, especially with reducing the error bars on these estimates. So the latest version looks like the RER is between 5 and 10 for the heaviest ions in this series. But the error bars are pretty large, of course. And also, the error bars and the values are determined to some extent by the model that's fitted. So the model that we used here was non-linear in general, but it can be approximated as linear at low doses when I showed that equation. So that has an influence as well on the estimates. But in general, yes, at lower doses, we estimate higher RER and RBE values than at higher doses.
If I could jump in
David, please.
just for a moment. Yeah, so a lot of this discussion really does depend on whether or not there is a different mechanism going on at very low doses, a non-targeted effect mechanism. And everything we've seen in whenever there's been some decent data at very low doses, there's good evidence, I think, for a non-targeted effect. So I think we have to think of, and the only way to get data for that is really to go to very, very low doses. So very hard experiments to do. But I think if using a dose response curve, which is basically the same at all doses, may be misleading us somewhat.
And let me throw in, what about low dose rate?
I'm smiling. Yes, yes, of course.
Okay, there's another question. This one's for Tatsu. Wow, Dr. Tatsu's graphs again indicated potentially very high RBE for very low doses in the milligrays of high LAT radiation. And I guess he's asking you to comment on that.
Yes, RBs are very high, but like expectable high in my opinion. So not too high, like 100 or 200. So like 10 or, and the things are very complicated because I analyze the tissue reactions, and in principle, like a zero grade never cause tissue reactions. So like very low dose RB is just by definition. So actually tissue reaction occurs at least like a 0.1 or 0.5 grade. So this is like an ideal value. So because of the model estimation. So maybe actual RB value is not so high, so up to 8 at the maximum. So compared to stochastic effects, RBs are not so high. So that's the important point for obtain from our study.
Well, I'm going to ask a couple of questions of each of you that are kind of related to what we're thinking about for the RBE report. So Igor and David, my question is, should RBE be totally replaced, with RR or something else? What are your thoughts?
Shall I have a go? Yeah, my thinking is that for the high dose scenario, which is mainly the clinical scenario, RBE is a better concept. It's what we want to know. What dose am I going to use to try and mimic the results I used to get? But for very low doses, I don't see any particular argument for RBE. And when I started to mull this over, I thought I just couldn't see anything that would support the use of RBE. Because that's really not what we want to know. We don't want to know the equivalent dose. We want to know how much bigger the effect is.
Yep. I think that makes a lot of sense. Igor, do you want to comment as well, or you let David talk?
Yes, absolutely. I agree that they can both be used in different situations where one is appropriate or the other one might be more appropriate.
And so then let me ask the second question, which is probably more directed at Tatsu, and that is, a lot of what we've done with RBE in the past has been about cancer endpoints, but you're leaning toward also looking at tissue reaction endpoints. But as you pointed out, tissue reactions are going to be more high dose responses than low dose responses. I personally believe it just is a measure of threshold, right? That there's probably a biology going on at low doses, but it's not enough to accumulate a tissue reaction. Do you think we should be considering RBE for high doses, for tissue reactions rather than just cancer?
I cannot follow the point. So high dose.
Well, you said, and I'm just, you had said that, and when I asked about the dermal effects, you said it's about high doses for tissue reactions, and you won't see much at low doses. So then I'm asking the question, should we be thinking about RBE for tissue reactions when it only occurs at high doses?
Well, that's hard to answer. So like a radiological protection purpose, maybe always like a conservative is an important point. So, but to prevent tissue reactions, maybe people are already exposed high dose. So in that case, like just conservativeness is not good, in my opinion. So try to estimate like more realistic values important even for the radiological protection purpose. So in that case, maybe I would say like roughly 2 to 3 grades, because for at least for skin reactions, some response occurs below 1 grade, but it's just I forgot the English name, but it's quite like easy to recover. So people do, particularly for like medical physicists, do not care about that. So what they really need to care is more serious damage, like discoloration. So I think it's okay to set a higher dose for like evaluating the recommended value. So that's my opinion. But we have to discuss a lot about this point.
Okay. There's another question from the other Tatsu Umeoka. Thanks, Tatsu Sato, for great talk. RBE for stochastic effects is greater than RBE for skin reaction, which is greater than cell survival. I guess that if radiation acts on 2 or more steps, which interact in a multiplicative manner, RBE can be greater. With an RBE of 2, the first step is twofold, and the second step is twofold. The result can be fourfold. Is this a plausible explanation? I think you can read it in the Q&A too, if it's a very long question.
Yes, I agree. Yeah. But it's not so simple, of course, but it's quite good way of thinking start. Yeah. And particularly to try to make like a evaluation case. So we have to introduce some assumptions or simplifications. So this kind of analysis is quite important. So I think it's quite interesting way to think about the ratio of RV for cell survival tissue reactions and stochastic effect. It's quite interesting. Thank you for Noka-san. Another Tatsuhiko.
Okay, I'm not seeing any other questions. I'm not seeing anybody else from the panel asking questions. Let me make a few comments. First of all, somebody asked about whether the slides will be made available. This presentation will be made available on video, so you'll be able to watch it again if you want, and ICRP will post that information. We normally try to keep these under an hour if we can in order to save people's time, so I'm grateful to our speakers for kind of sticking to time here. And let me thank everybody, including ICRP, for helping us to host this. Igor, David, Tatsu, this was really a very provocative work. I think it's going to help us think really hard about our next steps. So thank you.
And in turn, our thanks to you, Gail, for putting this together. Much appreciated.
Yes, thank you very much.
Thank you, everybody.